The central nervous system contains the spinal cord and brain. We will look at specific structures of both, the fluid-filled cavities of the brain called ventricles, protective structures, and the blood-brain barrier. We can compare and contrast the two organs of the CNS. Our brain contains the ventricles. The gray matter is superficial, while the white matter is deep in most brain structures. Those nuclei are gray matter structures housed within the white matter tracts. However, the spinal cord contains a central canal which is analogous to a ventricle. The white matter on this organ is superficial, while all the gray matter is deep. The spinal cord does not house nuclei, but contains gray matter horns and white matter funiculi instead. Our brains are divided into 4 major structures, each with fluid-filled ventricles. The cerebrum, responsible for your senses, conscious thought processes, somatic nervous control, etc. Diencephalon, homeostasis and autonomic control. Brainstem, reflexes, vital motor functions, and autonomic control. Cerebellum, balance and coordination and somatic control. Each major part of the brain has a ventricle. The ventricles contain structures that make the cerebrospinal fluid called choroid plexuses. That CSF is then circulated between ventricles, down the central canal of the spinal cord, and through spaces between the meninges that cover the CNS organs. Eventually, the CSF is then pushed to the blood supply for recycling. Cerebrospinal fluid serves as the transport medium for the brain and spinal cord. We also find it in areas where it provides cushioning between structures. The cerebrospinal fluid is made in a choroid plexus. Each of the 4 ventricles has a choroid plexus, which dangles down from the roof of the cavity. Capillaries are lined with ependymal cells, and these cells use osmosis to create the fluid. The ependymal cells also control the transport across the wall of the capillary and use their cilia to move the fluid. Use the free internet to research this condition and post your question and response to the ANP Cafe. We will now focus in on the cerebrum and its many tissues and structures. Our cerebrum is divided by the longitudinal fissure into 2 hemispheres. Shallow grooves, or sulci, separate ridges of tissues called gyri. The cerebral gray matter is also functionally separated into lobes. The cerebral cortex is amazing in its many functions. The ability to make synapses in the gray matter or integration result in feeling, talking, understanding, learning, remembering, and controlling skeletal muscles. The hemispheres are contralateral, and that they control or sense the opposite side of the body. We also have 3 general functional areas: sensory, receive internal and external body information and feel things. Association areas integrate that information in a synapse. And motor areas create the efferent commands to move somatic effectors. Here's an example. You take a bite of your favorite food. You taste that food in the sensory areas. Remember how much you love that food in the association areas, and move your arm to eat with the motor areas. On the cerebral gray matter, we do find specific functional areas. A handful of these are mentioned in your learning objectives. The lobes: parietal, which is somatic sensory; frontal, somatic motor and personality; occipital, vision; temporal, auditory; and sulla, taste. Within and between these lobes we find specific regions and gyri. The frontal eye field controls eye movements. Broca's area generates motor commands for speech. Wernicke's area understands spoken language. The ventricles: the cerebrum houses a lateral ventricle within each cerebral hemisphere. There are two substantial gyri on the human cerebrum on either side of the central sulcus. The precentral gyrus lies anterior to the central sulcus in the frontal lobe. Its gray matter generates somatic motor commands. We can see from the map where the specific body parts are controlled, and interestingly, the larger the body part on this map, the more control you have. Notice how large the tongue, face, and hand are. This implies that we have tremendous somatic motor control over those parts. The postcentral gyrus lies posterior to the central sulcus. This gray matter is somatic sensory and interprets sensory information for your skeletal muscles, joints, and the skin above them. Once again, we see the same trend in function versus the amount of gray matter devoted to that body part. Look at the huge size of the mouth, teeth, and lips here. The cerebral white matter lies deep. Here we find three tract types. Association fibers connect to gyri of the same hemisphere. Commissural fibers connect gyri between the two hemispheres, and so they cross sides. Projection fibers connect the cerebrum to an inferiorly located brain structure. Basal nuclei are the collected gray matter structures found within the white matter tracts. There are many basal nuclei in the cerebrum, and we don't expect you to know the names of all. These nuclei interconnect the cerebrum to other areas and perform specific function. Here we see some basal nuclei functions in red. Essentially, repeated behaviors get stored in these nuclei over time. When you were a child, someone taught you to tie a shoelace. The instructions were stored in a nucleus. As an adult, you simply access that information. No one needs to remind you now how to tie a shoelace. Also, when you first drove to campus, you might have followed a map. But after you learned the route, you no longer pay attention to the roads you take. Somehow, you get in that car and end up here. These are all basal nuclei functions. Influencing muscular activity. Repeated movements become memorized, as in tying your shoe. Attention cognition, your alert level. You may pay close attention to a task for the first 20 to 30 minutes. Some of us stay on task, others will have their attention wander. Inhibiting antagonistic or unnecessary movement, regulating somatic motor commands to accomplish a learned task. There are other functions. These are just a few. In blue, we see some disorders currently believed to have basal nuclei malfunction as part of the etiology. Not learning objectives, just interesting. Some of these pathologies involve attention span, muscular activities, and unnecessary movements. The diencephalon lies inferior to the cerebrum and contains 3 paired structures and the 3rd ventricle. The sides of the ventricle are formed by thalamus. Hypothalamus is the anterior floor of the ventricle. Epithalamus is the dorsal wall of the ventricle. The thalamus. These structures relay information from other parts of the brain to the correct part in the diencephalon. For example, visual information containing light are transmitted to the epithalamus. The thalamus have a lot of functions, including memory, sensory information, motor control, and regulation of sleep cycles. Hypothalamus is small but mighty. Its function is homeostasis, and we will dive deeper into that in a moment. The epithalamus contains the pineal gland. This structure is responsible for the sleep-wake cycle or circadian rhythm. Your pineal secretes the hormone melatonin at night, which tells the body it's time to sleep. Once the sun rises, the pineal stops melatonin production, and the falling levels are the body's signal to wake up. Back to the hypothalamus. Many of its functions are autonomic nervous system related, but it also has nuclei for emotional responses. Movement of your digestive tract, breathing responses to stimuli, organ movement, regulation of body temperature, heartbeat, and blood pressure are all autonomic motor functions. You can see emotional nuclei for pleasure, fear, anger, hunger, and satiety, your feeling of being full, as well as sleep cycles. Now, your hypothalamus is connected to the rest of the body by cranial nerves, but your hypothalamus also controls the pituitary gland. So we see that the hypothalamus is both nervous and endocrine body control. The pituitary gland contains 2 parts of different embryonic origin. The anterior pituitary developed from different embryonic tissue as the hypothalamus, and as a result, these 2 structures are connected by blood vessels. To control the anterior pituitary, the hypothalamus releases releasing hormones into that blood supply. Each hormone will act on target cells in the anterior pituitary, and they'll release a second hormone. For example, thyrotropin releasing hormone from the hypothalamus stimulates production of thyroid stimulating hormone from the anterior pituitary. The thyroid stimulating hormone will then act on the thyroid gland. The posterior pituitary, however, came from the same embryonic tissue as the hypothalamus. As a result, these 2 structures are connected directly with neurons. There are 2 hormones released from the posterior pituitary upon neural stimulation by the hypothalamus. Oxytocin should sound familiar as it is related to labor contractions. It also stimulates milk let down during breastfeeding, but also makes one person think another person is super cute and cuddly. Antidiuretic hormone is released at night, and while dehydrated, it stimulates water retention by the kidneys. Do not take notes on this diagram. This is so that you can appreciate the diversity in releasing hormones and the significant impact of the hypothalamus. The brainstem is divided into 3 structures with specific functions. Midbrain, the superior structure. Its nuclei control motor commands, sleep-wake cycles, and alertness. Pons, the sleep cycle, swallowing, breathing, bladder controls, equilibrium in hearing, taste, facial expression, posture, and respiratory rate. Medulla oblongata, also breathing rate, heart rate, blood pressure, digestive activities, sneezing and swallowing. The medulla oblongata also contains the crossover tracts for the body. Remember, the brain is contralateral. We have to switch sides somewhere, and the tracts going up and down to the spinal cord cross over here. The cerebellum is also large in humans. Our superficial cerebellar gray matter coordinates voluntary movements. It's also involved in physical balance and coordination, speech, and posture. Structurally, there are crossover tracts in the midline structure called the vermis and large cerebellar lobes that contain nuclei. Use the free internet to research this question. Post your response to the A&P Cafe. We've covered a lot of structures quickly. Take a few minutes here, pause the lecture, and review by answering these questions. In addition to these structures, there are functional systems. These are gyri, nuclei, or tracts that interconnect different parts of the brain for a specific function. The two main functional systems are the limbic and reticular. The limbic. This system contains the amygdala, cingulate gyrus, and hippocampus. These interconnect nuclei and gyrus provide emotional reactions and memories. For example, we connect memories to smell. I once had food poisoning and spent an evening vomiting near a trash can that contained mangoes, and for the longest time, the smell of mangoes made me physically sick. Our limbic system also helps us make physical gestures to reflect emotions as well. Reticular. This system regulates our attention span. The reticular activating system connects many areas to transition us from sleep to waking and to focus our attention. These diagrams show us the general locations of the two functional systems. Now, we're going to quickly discuss the functions of these structures. The hippocampus provides spatial navigation and long-term memory. Spatial navigation occurs when you instinctively back up to a chair and sit down on it without having to grab or look at it. Amygdala is emotional responses to smells and responses to fear or pleasure. Mammillary bodies, specific pieces of memories and smell. Olfactory pathways. Actually, far more involved than this. Many structures in our brains are associated with smell, learning smells, and understanding what smells mean to us. We store our conscious perception of smells in our frontal lobe, emotionally respond to smells with our amygdala and hypothalamus, and remember smells in our hippocampus. The cingulate gyrus links outcomes to emotions. Say you started walking every day and you feel so much better, the positive outcome motivates you to keep walking. Parahippocampal gyrus, part of the limbic. This gyrus pulls memories from other areas. You can practice these structures and functions on this Quizlet set. The human central nervous system organs are the texture of room temperature butter, so kind of soft. Also, we've learned that neurons are quite sensitive. This implies that the brain and the spinal cord need protection. Bones, meninges, cerebrospinal fluid, and the blood-brain barrier do just that. You've already learned that the skull sutures don't easily move. This makes a solid shell of bone around the brain and a flexible series of bones around the spinal cord, definitely protective. Just under the bones are 3 tissue layers, collectively called meninges. Not only do these cover the organs, but they also house a fluid-filled cavity, the dura mater. The most superficial tissue that's somewhat tough and bound to the periosteum of the bones in the cranial cavity, subdural space. This is a potential space that shows up with cranial trauma, not typically visible. Arachnoid mater, named after its spiderweb appearance, this layer houses a permanent cavity with cerebrospinal fluid and vessels. Subarachnoid space, the permanent cavity where blood vessels and cerebrospinal fluid are both found. This provides a fluid-filled cushion around the brain and spinal cord. And pia mater, the thinnest and innermost meninge that is bound to the brain and spinal cord. Note, these are the cranial meninges. The spinal meninges are essentially the same. However, there is an extra epidural space filled with adipose tissue just inside the vertebra. Then we would find the dura mater, etc. We've seen that the cerebrospinal fluid is created in the ventricles, flows down and out the spinal cord and around the organs. This fluid must eventually be pushed back into cavities for collection by blood vessels to maintain fluid balance. There are valve-like structures that allow cerebrospinal fluid to come out of the subarachnoid space and collect in very specific places for reabsorption to the blood supply. Those structures are called arachnoid villi. Blood does not come in free contact with the nervous tissues. Whenever blood vessels are associated with the nervous system, a blood-brain barrier will be in place. This barrier has 3 parts. A continuous endothelium. This is the lining of the capillary wall. This type of capillary is permeable to only small molecules anyway. The basal lamina is the membrane of the endothelium, and fused to this membrane are glial cells. Astrocytes and ependymal. Depending on where the blood vessel is, we'll find glial cells wrapped around it. These cells will control the kinds of molecules that enter and exit the blood supply. The blood-brain barrier is everywhere, except in these places: hypothalamus, medulla oblongata, pituitary gland, pineal body. This is because these 4 areas release hormones or monitor blood chemistry. Therefore, the neurons here do need direct access to blood supply. Not learning objectives. Any compound that's permeable across the blood-brain barrier will cross and affect neuron function. That's exactly how medications, drugs, nicotine, and alcohol work. These molecules cross the blood-brain barrier and change neuron function. Just Prof. Klein being funny, we're switching gears and going to touch upon memory.
information is stored within synapses of different brain structures. Now, if I ask you about the same subject in 6 months, you are very familiar with it and can explain the major themes. Long-term memory is what you want to achieve in this class. As your future program will assume, you have mastered the lecture and laboratory content. We have seen sections of the list. The amygdala, hippocampus, and cerebral cortex are all memory areas. Now here we see the 3 stages of memory. Encoding, your initial interaction with a piece of information is sensory input. Storage, the different synapses in short and long-term areas. And retrieval, when you pull that information out of storage or the act of remembering. For example, you first heard about cranial nerves in class during encoding. If you reviewed those cranial nerves a few times a day for a few days straight, you begin storing the information in synapses. When you sit for an exam, because you have continuously reviewed, you retrieve the information. This information is not in the learning objectives and again presented because it's an interesting execution. If you look at the 3 brain scans, you can easily see large dark areas in the Alzheimer's brain. This condition is progressive and results in a personality change followed by memory loss. There is hope, however, as multidisciplinary treatment approaches seem to slow the progressive loss. In some research fields, Alzheimer's is referred to as type 3 diabetes, and that's obviously controversial. So again, we might have a pathology example that is blood sugar related. Might. Since there's so much content in this unit, we may use these favorite flashcards to review. Well, welcome to the cranial nerve club. Yes, we are going to discuss our cells. These serve nerves come off the bone. Some are purely sensory, others are purely motor, and some contain both fiber types. Of the 12, there are 4 with parasympathetic fibers. What did you learn here for lecture? The name, type, and functions of each. Eye nerve achnotting. The macroscopic second nerve is that with a pointed to the teeth called the epimerium. Within the nerve, we find bundles of myelinated axons, or vesicles, that are bound by perimerium. Lastly, if we enter a fascicle and pull out one nervous fiber axon, we will find it bound by endomerium. This chart serves as an overview. You can see each nerve in numeric order, the type, pathways of taste. Motor is spatial expression and salivation. Cranial nerve cell 8, vestibulotrochlear, is sensory, hearing, and balance. Cranial nerve cell 9, glossopharyngeal, is mixed fiber. It carries parasympathetic, the sensory or blood chemistry, and blood pressure. Motor is muscles for swallowing in the GAD reflex. Cranial nerve cell 10, vagus, is a mixed nerve, which contains parasympathetic fibers and provides parasympathetic control.